Thank you. Welcome, everyone. My name is Rita, and I did my master thesis at the Research Institute in Bergen. My main topic was the in vitro digestion method for assessing amino acid digestibility of black soldier fly larvae for salmonids. So, aquaculture has been one of the fastest growing industries of the last 3 decades. And as we can see here in this graph, we can see the aquaculture for human consumption. And we can see that's growing exponentially compared to the total capture fisheries and the capture fisheries for human consumption. We're getting now to a point where the aquaculture for human consumption is surpassing, and it's continuing to grow until 2030. So considering the continuous growth of the world population, the demand for food, and the decrease of the natural fish stock, the aquaculture sector will most likely continue to expand. So since this sector continues to expand, a lot of effort was put into making aquafeeds for fish, and in this case for salmonids. I have here a graph that shows us the composition of the aquafeeds for salmonids, and we can see here in purple and blue the fish meal and the fish oil. Overall, they constitute 29% of the percentages of the meals, and however, this is not very good because the limited marine resources that we are now facing because of the overfishing of the oceans, and also in here in this graph we can see the plant protein and the plant oil, that it's more than 30%, and the plant-based ingredients mostly are based on soy. They raise a lot of environmental concerns, especially in the places where they are produced, like the Amazon forest. So we still need to feed our salmonids. We still need to have an aquafeed that meets the requirements of the salmonids. So we need we have the need for new feed ingredients, and these new feed ingredients could be insects. Insects have been studied a lot in the last few years since they were allowed in the European Union. A very important insect that's been used is BSF, the black soldier fly larvae, which I'm going to focus on this presentation. And we know that they are pretty good because they have between 30 to 58% of protein. They have a very good essential amino acid profile. They have a very good mineral source. However, they lack in a good fatty acid profile for salmonids. So we need to increase this insect meal quality by processing it in a process called defatting. So we have these new feed ingredients coming in, new sustainable feed ingredients coming in with the new processing methods. So we need to create a new screening methodologies that will allow us to quickly go through these ingredients and see if they are good. And these new screening methodologies are called in vitro digestion methods. An in vitro digestion method, it's basically a preliminary assessment of the quality and performance, like the digestibility of novel feed ingredients, like the insects, before their inclusion in the aquafeeds. And this nutritional evaluation of novel ingredients normally is done by in vivo methods, but these in vivo methods can be very long, they can be very expensive, and unfortunately they involve a large sacrifice of animals. And we have, on the other hand, an in vitro digestion method that's less time consuming. In 2 days, you can go through the whole protocol. It's a lot cheaper, and there's no direct use of animals. So in this thesis, in order to assess the amino acid digestibility of BSF meals for salmonids using an in vitro digestion method, a main research question was proposed, which was which digestion factors influence the amino acid digestibility of BSF larvae meals. So first, we optimize an in vitro digestion method. This is the first part of my thesis. In the design of the method, we started with a two-step digestion process where we started with an acidic hydrolysis, where we chosen the pH in the stomach, which was 2 or 4. We know that according to research that in salmonids, the pH in the stomach goes between 2 and 4, which shows the reaction time in the stomach 1 or 2 hours. We know that evacuation of the stomach happens 2 hours after the ingestion of feed. And the concentration of enzyme in the stomach was 1 unit per milligram of protein, or 7 units per milligram of protein. And we also, according to research that we did, that other in vitro digestion trials used until 25 units. And we decided to use a very basic one, the 1 unit, and then have a more intermediate one, the 7 units, to test the range of concentration of enzyme. And then we have an alkaline hydrolysis, which constitutes in the pH in the intestine, which was 7 or 9. Also in here, according to research, that's how we chose these values. The reaction time in the intestine was 2 and 4 hours. We chose these values because, according to literature, the first material that appears in the hindgut appears 6 hours after feeding. So this follows a logical time frame. And then the concentration of enzyme in the intestine was 1 unit or 7 units, and we follow the same logic as the one in the stomach. And finally, the temperatures that we worked with was 12 degrees and 37 degrees. 12 degrees because we know in Atlantic salmon that the optimal growth and ingestion occurs at 12 degrees, and 37 degrees since we used porcine enzymes. They come from mammals, and according to product description, they work better at 37 degrees, so we also use this to control and check. So overall, this design was a fractional factorial design, and overall we had 32 experiments. So of all of the 7 factors, we combined them in 32 experiments. So in here I present you a flow chart of the method that we developed. So we used a BSF 0 meal, which is a partially defatted BSF protein meal. We weighted 0.2 grams of the sample, as you can see in picture number 1. Then in picture number 2, it shows the acidic hydrolysis, where we pipetted 2 milliliters of acid solution and we added pepsine. The samples were put in rotation in an appropriate temperature. Then alkaline solution was mixed and the protease, chymotrypsin, and trypsin were added to these tubes. The samples were put in rotation again, like you can see in picture number 3. Then we collected the tubes, we centrifuged the samples, and we divided in a non-soluble fraction and a soluble fraction. And then the non-soluble fraction was analyzed by the UPLC method. And basically what we did, we calculated the digestibility, and then we subtracted to 100, because in the soluble fraction we have the product that was not digested. So in the results of this method, in here we have a graph with the experiments in the x-axis, and the y-axis we have digestibility, and in here we have the amino acids with represented by the different colors. And we can see that in experiment number 11 it had the highest digestibility of all of them at 37 degrees, and the same happened in the non-essential amino acids. And we also have the highest digestibility in experiment number 11. So you might wonder, what is experiment number 11? So it's several factors put together. So the pH in the stomach used here was 2, the most acidic one. The pH in the intestine was 9, the most alkaline one. The reaction time in the stomach was 1 hour. The reaction time in the intestine was 4 hours. The concentration of enzyme in the stomach was 1 unit milligram of protein. And the concentration of enzyme in the intestine was 7 units of milligram of protein. And the temperature was 37 degrees. So these ones were the optimal conditions. So after we optimized the in vitro digestion method, we reached the most optimal conditions, and we decided to answer to 2 more questions. Can we use this in vitro method to evaluate differences of amino acid digestibility between batches? So batches that came from the same company but produced in different months. And can we use this method to evaluate if the processing of BSF meals affects the amino acid digestibility? So to answer the second question proposed, We used what we call the BSF 0, 1, 2, and 3. All of them were partially defatted BSF protein meals coming from the same company. In here we have a heat map where we looked at the digestible amino acids. We have all of the essential amino acids tested and the BSFs in the bottom. And at a darker green, the highest digestible amino acids, and a lighter green, we have the less digestibility. And we saw, like in here in methionine, that BSF0 and BSF2 were significantly different of BSF1 and BSF3. We checked this by doing a one-way ANOVA and a Tukey test. And here again, the non-essential amino acids all present in the y-axis and the meals in the bottom. And we could also see, according to the statistics analysis that we did, that the digestible amino acid values of essential and non-essential amino acids were almost significantly different. So this could be due to several factors. It could be a human error in a method, natural variation of the insects provided, different rearing substrates of the insect, or maybe some differences in the defatting process. So now quickly passing to our third question, which was application of in vitro method to different processed meals. We tested 5 different meals. BSF4 was a defatted protein meal, BSF5 was a normal protein meal, 6 was a whole larvae meal, 7 was a protein meal too, and 8 was a defatted protein meal. And in here, we have another heat map with the essential amino acids presented, and in the bottom we have the BSF0, then BSF4, 5, 6, 7, and 8, and we saw that overall methionine had the lowest digestibility, and also it's a limiting amino acid, so it makes sense. In here, in the non-essential amino acids, we used as a comparison, and we saw that BSF4, which was a defatted BSF protein meal, as you can see here, and BSF number 5, which was a normal protein meal, had the highest digestibility values. The BSF meals that were partially defatted, like BSF0, and completely defatted like BSF4 had higher digestibility than BSF6 that corresponds to the whole larvae meal, which had no type of processing. So actually processing helps with the digestibility. So to conclude, we draw 3 different conclusions. The first one is in the optimization of our method. We came to the conclusion that the best conditions were temperature at 37 degrees with a pH 2, reaction time 1 hour, and 1 unit of pepsine per milligram of protein during the acidic hydrolysis. And when the pH was 9, reaction time 4 hours and 7 units of protease, chymotrypsin and trypsin during the alkaline hydrolysis. The second conclusion that we reach is that BSF meals from different batches and from the same company overall had significant differences in almost all essential and non-essential amino acids. And conclusion number 3 is that BSF4 and BSF5 had overall the highest digestibility, this corresponding to the defatted BSF protein meals comparatively then to the whole larvae meal. So this method, there's still a lot of things that can be done. We believe that further validation needs to occur to correctly assess other insect ingredients, plant-based ingredients, and fish-based ingredients. So there's a lot of room to improve in this area. Use this in vitro digestion method to assess other nutrients like minerals and lipids for salmonids would be also a great idea. And finally, a comparison between an in vitro study and in vivo data for a possible correlation between the 2 methods. This could give us a more insight about what's happening and if a possible correlation could be useful. So, yes, I would like to thank you all for your attention. I would like to thank my supervisors, Ikram, Martha, and Anthony, for helping me out throughout this whole process. I would like to thank Pedro that helped with the statistics analysis of this thesis. I would like to thank you the all IMBRSea community and the requirements and welfare group at AMR. Thank you so much for your time.